Hello friends, welcome to BMH Learning. This video is about Bile Esculin test. The Bile Esculin test is a biochemical test performed to differentiate Enterococci and group D Streptococci from non-group D viridans group Streptococci on the basis of their ability to hydrolyze esculin. Many organisms are capable of hydrolyzing esculin, but only a few of them can do so in the presence of bile, that is 4% bile salts or 40% bile. Thus, this property is utilized to identify organisms of a particular group. The bile esculin test is performed on a selective differential agar, bile esculin agar, which consists of bile as well as esculin. The agar contains different bile salts that inhibit the growth of other gram-positive organisms and allows the selective isolation of Enterococci and group D Streptococci. Principle: the basis of the esculin test is the hydrolysis of esculin in the presence of bile salt as a result of enzymatic action of esculinase. Esculin is a glucoside consisting of glucose and hydroxycoumarine linked together by an ester bond through oxygen. The bile esculin test selects organisms first on the basis of their ability to grow in a medium with 4% bile salts, followed by the selection based on their ability to hydrolyze esculin. The hydrolysis of esculin results in glucose and a compound called esculatin. After the degradation of esculin, the esculatin produced by the hydrolysis of esculin reacts with iron ions in the medium to form a phenolic iron complex, resulting in a dark brown or black color. This is the list of the chemicals and the other requirements you need to carry out this experiment. You need to use the chemicals as mentioned. Procedure: preparation of media. In a beaker, 64.5 grams of the dehydrated powder is added to 1 liter of deionized or distilled water. The medium is then heated up to boiling to dissolve the powder completely. The dissolved medium is then distributed into tubes and sterilized in an autoclave. Once the autoclaving process is complete, the tubes are taken out and cooled at a slanted position to a temperature of about 40 to 45 degrees centigrade. The position should be maintained in order to obtain butts of 1.5 to 2.0 cm depth. Esculin hydrolysis. Esculin hydrolysis can be observed either through a tube test or a disc test. In this video, we will talk about tube test. A well-isolated colony is taken from 18 to 24-hour culture with a sterile inoculating needle. The bile esculin agar tubes are inoculated by streaking the surface of the slant with the light inoculum picked from the culture plate. For Enterococcus and S. bovis identification, 40% bile is used, and the tubes are inoculated with a 10 microliter calibrated looful of a 0.5 McFarland standard suspension prepared in sterile water. The cap of the test tubes should be left loosened to ensure adequate aeration. The tubes are then incubated aerobically at 35 degrees centigrade for 24 hours, and the color change is observed. Result interpretation: a positive tube test in medium containing ferric ammonium citrate is indicated by the blackening of the medium. A negative tube test is indicated by a lack of color change. The medium will fluoresce under UV light 366 nanometer. This was all. Thanks for watching.